cholesterol metabolism.
The topic included in this presentation is how does fasting regulate cholesterol metabolism?
Back to the regulation of cholesterol synthesis, we have the hormonal regulation. After meals, insulin induces activation of hydroxymethylglutarate CoA reductase by activating a protein phosphatase that dephosphorylates the hydroxymethylglutarate CoA reductase with subsequent activation and increased cholesterol synthesis. So, as we see, feeding promotes cholesterol synthesis by providing the substrates for cholesterol synthesis, which are acetyl coenzyme A and the reduced NAT phosphate, and activation of the regulatory enzyme hydroxymethylglutarate CoA reductase. During fasting and the periods of food deprivation, we have glucagon, the hormone that activates adenyl cyclase with increasing levels of the cyclic AMP and subsequent activation of a protein kinase that phosphorylates the regulatory enzyme hydroxymethylglutarate CoA reductase and inhibits its activity. Subsequently, cholesterol synthesis is reduced. This process is AMP dependent, so when ATP is reduced, there will be no need for cholesterol synthesis. So, as we see that fasting is a good cholesterol lowering strategy where the hydroxymethylglutarate CoA reductase activity is reduced. So, as you see, the covalent modification of the enzyme is key for regulating cholesterol synthesis. The hydroxymethylglutarate CoA reductase is covalently modified by phosphorylation-dephosphorylation reaction. The active form is a dephosphorylated form. This is achieved by a protein phosphatase that is activated by insulin, whereas it is inactivated by phosphorylation by a protein kinase, which is activated by the hormone glucagon during fasting and starvation. And hence, this explains the role of fasting in down-regulating enzyme activity and cholesterol level reduction. To summarize the previous regulatory mechanisms for the activity of hydroxymethylglutarate CoA reductase, we have short-term regulation by phosphorylation, dephosphorylation, and long-term regulation by proteolysis of the enzyme itself and cholesterol-stimulated degradation of the enzyme. Also, gene expression and regulation by the steroid sensing domain and the strip protein. If we understood the previous points, we can now explain how does fasting inhibit cholesterol synthesis and explain how the feeding fasting cycle regulates the cholesterol metabolism. One more question before the previous two questions. We can explain how carbohydrate-rich diet leads to cholesterol synthesis and hypercholesterolemia.
If you like this lecture, please hit the subscribe button and give your feedback in comments. The link for this lecture will be in the description below this video, and you can find other details in the website provided.